Hello children, welcome you all to the Pearson edXL Form 7 online biology class. Today I'm going to discuss one of your third midterm lesson, Decomposition and Forensics, which comes under the SUW reference topic 6C, page number 146 to 158. During this lesson, you will be able to understand the process of microbial decomposition of organic matter, how to determine the time of death, and the cutting-edge technologies of genetic engineering, the PCR and gel electrophoresis. Okay children, first of all let's move towards the microbial decomposition. Microorganisms such as bacteria, fungi, they are playing an important role in breaking down of complex organic matter into simple organic matter. They are important for nutrient recycling. So the process decomposition is consisted of two main parts, autolysis and putrefaction. Autolysis means where the body's own enzymes digest and break down tissues. Putrefaction means where microorganisms such as bacteria and fungi, they are breaking down the remaining dead tissues. Now in this diagram, you can see the carbon cycle. How carbon is being recycled in the environment. So basically the dead organisms and the waste product are decomposed by the activity of the microorganisms. So during this process, carbon dioxide is being released. So further, this atmospheric carbon dioxide can be absorbed by the plants. So now you can see the summary of that diagram. Microorganisms decompose organic matter and the simple organic matter is released to the soil, where these are utilized by plants. During the decaying process, carbon dioxide gas is released to the atmosphere. This released carbon dioxide will be absorbed by plants via the process called photosynthesis. These plants fix carbon dioxide to produce carbohydrates. These carbohydrates are moving through the food chains and food webs and eaten by consumers. That is how the carbon is being recycled. In the same manner, nitrogen can also be recycled. Phosphorus can also be recycled. Okay guys, now let's see what is meant by forensic science. In this major, you'll be able to reconstruct crimes by analyzing physical evidences. What are the physical evidences? Blood, DNA, fingerprints, and some clothing materials. So these evidences can be used in a court of law. People die. Sometimes they are killed, unlawfully or murdered. It is not a surprise that these crimes have a high profile in the media. So, to secure prosecution for murders, number of facts are to be verified. What are these factors? Who died and who killed? What are the physical evidences? Where and when this murder took place? So we have to consider these factors. And also children, when investigating the time of death, these are the facts to be considered. So now let us see how to estimate time of death. How long an organism has been dead can be estimated by stages of succession, body temperature, degree of muscle contraction, we call it rigor mortis effect, forensic entomology, and extent of decomposition. First of all, we'll discuss about the stages of succession. In here, there are 5 main stages of decomposition: fresh, putrefaction, fermentation, dry decaying process, and the decaying of the skeleton. We call it skeletonization. that occur after death, each attracting different types of organisms. They are coming and feeding on the dead body. So the different stages can occur over days, weeks, and even years, meaning analysis of succession can aid estimating the time of death of even long-dead organisms. A pattern of insect succession results as different carrion insects are attracted to the varying biological, chemical, and physical changes of the dead body, which is undergoing through decaying process. Okay, children. Now it is about the body temperature. So what happened to the body temperature when a person dies? The body temperature decreases over the first 24 hours in the following shaped curve. So gradually the body temperature decreases. The environment the body was found in must also be considered. Since it found in water or where there is air movement, it speeds cooling, while clothing and being indoors slows cooling process. Right, now we are moving towards the degree of muscle contraction. We call this rigor mortis effect. When someone dies, heart stops pumping blood. When you are alive, your cells can respire aerobically, while after death, muscle cells which have stores of ATP continue to respire anaerobically, and this makes muscles more contracted. So what happens? The body becomes rigid. This is called as rigor mortis effect. On average, this starts about 2 to 4 hours after death, and this is taking place between 6 to 8 hours for full effect. Now in this slide, children, you can see some of the diagrams that show stiffened muscles in dead bodies. Right. Next one is forensic entomology. So this means the study of insects found in the body. So here in the diagrams you can see green bottlefly, blowfly, so larvae of blowfly and carrion. These are the insects which are attracting to dead bodies, and they are feeding on these dead bodies. So here the maggots can also be used by observing their length and the temperature they are found at, and also looking at what stage of their life cycle they are in. So these things can be used to estimate the time of death of a certain person. Now this is the life cycle of blowfly over time on dead bodies. So if we found the body after like 23 hours, we can observe the first larval stage. After like 130 hours, what we can observe is the pupa. Right? So likewise, we can use the stages of these organisms to estimate the time of the death. Right, children. The last one is the extent of decomposition. Simply, this means the appearance of the body that can give an estimate to time of the death. So here I used one example how the discoloration of the abdomen happens. So greenish discoloration of the abdomen can be observed after the 36 hours of death. So then it is spreading across the rest of the body in between 36 to 72 hours. So after 72 hours, so this discoloration further increase and it can be further turned into dark purple color. And also finally, if it is like 1 week, the body becomes bloated with gas. And also children, now let's see this diagram. So the first diagram shows a closed stomach. Second one shows an open stomach of a dead person. So hours of last meal before the death can be used to estimate the time of death. So in here you can see semi-digested food particles in the stomach. Alright children, so as you have seen so far, there are so many cutting-edge technologies in genetic engineering. Right? So they are genomics, proteomics, and This PCR technology is gel electrophoresis, right? So first of all, can you please tell me what is the genome? Genome means the total genetic materials in an organism. In DNA sequencing, we analyze individual strands of DNA, giving us the patterns of bases that code for certain proteins. In criminal investigations, there may be small DNA samples available. So, we are using this PCR technology make it possible to produce enough DNA sample for our analysis. Right? So, let's see how this PCR can be done. Now, these are the PCR components. So, first of all, we are taking the DNA sample and then it is mixing with primers, nucleotides, buffer, and Taq polymerase enzyme. So here this Taq polymerase enzyme is extracted from Thermus aquaticus bacteria. We have extracted this Taq polymerase enzyme from a thermophile. Thermophile means the bacteria who can survive under extreme temperature conditions. Because in here we are increasing the temperature. As we all know, the enzymes are getting denatured in high temperatures. But Taq polymerase does not denature in high temperatures. That is why we are using this enzyme. So PCR process is consisted of three major steps. First one, denaturing. Second one, annealing. Third one, extension. So during the first stage, we are increasing the temperature into 95 degrees. So the DNA double helix separates from each other. Then the annealing process. So the primers we use are binding with the corresponding strands. Then they are starting to amplify, further increasing the temperature to 72 degrees. So this 72 to 75 degrees is the optimum temperature for Taq polymerase enzyme. So these steps are repeated around 30 times, producing about 1 billion copies of original DNA. So the equipment that we use to perform this PCR test, thermal cycler, or we call it PCR machine. Okay, now we are coming to the topic DNA fingerprinting. So what is this DNA fingerprinting? It means the genetic fingerprints that are unique because people have different number of VNTRs. What are these VNTRs? Variable number tandem repeats. So these repeats of certain sequences of bases, it can be vary from person to person. For an example, one person may have 10 repeats of CAG base sequence over and over again. But same sequence can be repeated like 50 times in another person at various positions across their genome, making parts of their DNA different lengths. Right? So because of these, two individuals never get the same DNA fingerprinting. So comparing these genetic fingerprints can be used to determine genetic relationships. For an example, paternity testings, maternity testings, to identify suspects of a certain criminal cases. Right? So always the chances of two individuals having the same VNTRs at all locations is so slim. Alright children. So before we are moving to the gel electrophoresis, I would like to ask one question. How is a DNA profile produced? First of all, DNA should be fragmented using restriction in the nucleus enzymes. Then after they should be separated and we can perform gel electrophoresis to identify the banding patterns. So this whole process starts with the gel electrophoresis. So basically the gel electrophoresis is a process used to separate DNA fragments according to their size using an electrical current. So these are the steps. First of all, the amplified DNA is placed in a slab of gel smothered in a conductive buffer solution through which a current is passed. Then after we all know DNA is negatively charged. So DNA is attracting towards the positively charged anode. So shorter fragments of the DNA, they are traveling much more faster than the bigger fragments. So the position of DNA across the slab is unique for each person. So this is simply called as the genetic fingerprint. So here now you can see the gel electrophoresis apparatus. You can see the cathode, anode, which are connected to the power supply, and the agarose gel. And there are wells dipped closer to the cathode. We can inject samples for these wells. So these DNA fragments, when they are moving, they are attracted towards the anode. So based on the size, they can be separated. So in this diagram, you can see how to inject samples to the wells of the gel. So this is the banding patterns of gel electrophoresis. Okay, that is the end of the presentation. Before the end, I would like to recall the things that we have done so far. We discussed about the process decomposition, how to estimate the time of death, the PCR, and DNA fingerprinting and the gel electrophoresis process. We have discussed these things. Now let's move towards the questions. Okay, now there is a big problem. There is a mother and a child. But we want to find who is the true dad of this child. So what we can do is we are performing the DNA profiling and gel electrophoresis for all these people, for mother, child, and for 3 male persons. Right? Then what we do is we are just comparing the banding patterns of the child with this DAD1, DAD2, and DAD3. All these patterns are not exactly similar, but some bands are matching with the correct dad. So by comparing these 3 dads profiles, we can come to a conclusion that one has the most matching pairs with the child. So who is the true father now? Dad 1. Likewise, you can do the same thing for question number 2 and for question number 3. Who is the suspect of this crime? You can try this at home. Okay, children. Now there is another question. So you have to refer the page number 155 of your textbook and try the questions given about DNA profiling, PCR, and gel electrophoresis. That is the end of the presentation. I hope you all enjoyed. Thank you for listening.